Hello students, today we'll learn again a new concept, or we'll say a very important concept in molecular biology, and that is central dogma of protein synthesis. Central dogma of protein synthesis or central dogma of molecular biology. It is also called as central dogma of life, because in every living organism, the protein is synthesized in a same manner, in a same unique manner. Okay. Now, in the last lecture, we have talked about the structure of DNA. Now, DNA is a double-stranded structure. Which is made up of nucleotides. And a segment of a DNA, it is called as a gene. So, if you want to define gene, what is a gene? Gene is a segment of DNA which will express itself in a form of a protein. Gene is a segment of DNA which will express itself in form of a protein. Okay. Now, we also know that human being, there are 46 chromosomes. And these 46 chromosomes, we call it as a diploid set of chromosomes, that is 2n. And when a cell, 46 chromosomes, and now that is present in every cell. Chromosomes are present in every cell. So let me draw a cell for you. This is a cell, and here I said human being has 46 chromosomes. Now these chromosomes are present in every cell of your body. These chromosomes are every cell, wherever you know that the living organism, all the basic unit of life, it's called as a cell. And every cell has a whole set of chromosomes. There are exceptions, that is gametes. Gametes will have haploid set of chromosome, and other somatic cells will have diploid set of chromosome. So these chromosomes will have genes, and these genes will encode some proteins. What is a protein? Now, protein is a biomolecule. A protein is a biomolecule. It is made up of amino acids. Protein is a biomolecule. It is made up of amino acid, that is a polymer. Which is made up of amino acid, and we know that there are 20 amino acids which are coded, which will form this protein. Now, when you talk about 46 chromosomes present in every cell of a body will not make all the proteins required. For example, the salivary glands will secrete saliva. The sweat gland will secrete sweat. Okay. So, whatever function a cell has to perform, the cell will perform only that particular function. That means the synthesis of protein is highly regulated. What I mean to say is, a cell which is present having the whole set of chromosomes will have the whole set of genes, but will synthesize only required protein. The physiological role of every cell is defined, and the kidney will have its function, the liver will have its function, the heart will have its function, and every cell will have its own function. And the functions which are played by the protein are very specific and unique for every cell. Okay, so this synthesis of protein is highly regulated. The synthesis of a protein is highly regulated. Okay, now when you say the cell, where is this protein synthesis carried out? It is carried out in the cytoplasm. In the cytoplasm, that there are ribosomes. There are ribosomes, okay, and this is the messenger RNA. This is the messenger RNA. Now, messenger RNA brings the information from the DNA. As I told you, every cell has chromosomes, and the chromosomes will have DNA. The chromosome is made up of DNA. So, the information to synthesize a particular protein is brought from the DNA. Okay, the information to synthesize a protein is brought from a DNA. Now the scientist name Watson and Crick, who gave the structure of DNA. Okay, Watson and Crick. Now these are the scientists who have given, two scientists who have given us the structure of DNA. They said that whatever protein has to be synthesized in a cell, the information, the information has to come from the DNA. Okay, I mean to say, whenever a cell synthesizes a protein, the information has to come from the DNA. This was suggested by Watson and Crick for the first time, and they have even published it in the research journal. Okay. Now this information that is from the DNA, the message should go to RNA, and from RNA the protein will be synthesized. Okay, so DNA, it is a genetic material. The information is passed, the genetic information, DNA is a genetic material. The information is passed to the RNA in the form of messenger RNA. Okay, so from the cell, let me rub this. So from the cell, the nucleus, the DNA is present in the nucleus. From the nucleus, the information is brought to the cytoplasm. This is the cytoplasm of the cell. Information is brought to the cytoplasm, and the protein synthesis is carried out in the cytoplasm of the cell. The protein synthesis is carried out in the cytoplasm of the cell. Okay. So, from the DNA, when RNAs are formed, it is called as transcription. And from messenger RNA, when proteins are synthesized, it is called as translation. Okay. So, when the message or when the genetic flow, the genetic information is transferred from messenger RNA, from DNA to messenger RNA, it is called as transcription, and from messenger RNA, when the proteins are synthesized, it is called as translation. Okay, this is called as central dogma of protein synthesis. It is called as central dogma of protein synthesis. Central dogma of protein synthesis. Or it is also called as central dogma of molecular biology. Okay. Now this flow of genetic information is unidirectional. The genetic information will always flow from a nucleic acid to a nucleic acid, and from the nucleic acid to protein. Okay, the genetic information can never flow from the protein to nucleic acid. It cannot flow from the protein to nucleic acid, and hence it is called as unidirectional. It is called as unidirectional. The genetic flow is always unidirectional, that is from DNA to mRNA and from mRNA to protein. From mRNA to protein. Okay. Now when a cell divides, okay, when a cell divides, it always has to have its whole set of chromosomes. Okay, suppose this is a somatic cell and it is undergoing a mitotic division, which will divide into 2 cells. Again, it will divide into 4 cells. So every time it divides, it should have the whole set of chromosome, and hence the DNA needs to replicate all the time, and that is called as DNA replication. It is called as DNA replication. Okay, so when the DNA replicates, there are a lot more enzymes which are involved in the DNA replication, and there are different types of DNA replication. Okay, so DNA replicates into its own kind, and then when it is transcribed from DNA to mRNA, the double-stranded structure changes to a single-stranded structure. It changes to the single-stranded structure. The DNA is changed to mRNA. Okay, it is converted into mRNA, and then protein synthesis, that is a polypeptide chain, a single polypeptide chain is formed. Okay, this is the central dogma of protein synthesis. Now we know that DNA is a genetic material in almost all the living organisms, whereas in some viruses, RNA is a genetic material. Now, for example, HIV virus, COVID virus, they have RNA as a genetic material. So, in the animals or in the living organism where DNA is a genetic material, the flow is like that, that is DNA to mRNA and to protein. Okay, but in case when the genetic material is RNA, the protein cannot be synthesized from RNA to protein. Okay, only from RNA the proteins cannot be synthesized. The proteins cannot be synthesized only from RNA. And then what happens? It has to convert to DNA. It has to convert to DNA. Okay. So what I said is, in few viruses where RNA is a genetic material, where DNA is absent, RNA is the genetic material, and when they infect any host cell, they need their proteins to be synthesized, but on their own they cannot synthesize their proteins. So what will happen? The virus will die. Okay, they cannot replicate, they cannot multiply. So when they enter the host, when the viruses enter the host, from the DNA, from the RNA, they make their DNA, and this process is called as reverse transcription. It is the reverse of transcription, and hence it is called as reverse transcription. It is called as reverse transcription. So from RNA, when DNAs are synthesized, it is called as reverse transcription. Okay, and these viruses will have an unique enzyme that is called as reverse transcriptase. The enzyme called as reverse transcriptase will be present in this in these viruses. Reverse transcriptase. Reverse transcriptase will be present, and in this condition RNA can be transcribed to DNA. Okay, and the process is called as reverse transcription. The process is called as reverse transcription. Okay, so what we have done, So what we have studied today, that is the central dogma of molecular biology. Central dogma of molecular biology, what it suggests is every time when a protein has to be synthesized, the flow of information, the genetic flow of information should be unidirectional. That is, from the DNA, the information is passed to the messenger RNA, and from messenger RNA, it should be coded into proteins. And we also know that proteins are made up of amino acids, and they are biomolecules, or we say they are polymers. Okay, the information flow is always unidirectional. That is, from the nucleic acid will synthesize, but the formation of protein to nucleic acid is not possible. Okay, now this reverse transcription, that is in case of viruses where the genetic material is RNA, reverse transcription is possible, and this was given by a scientist named Temin, and hence it is called as Teminism. Temin and Baltimore. These are the scientists who has given the concept of, who have suggested that reverse transcription is also possible, and this is a modification of central dogma of protein synthesis. It is a modification of central dogma of protein synthesis. Okay, so from RNA, when DNA is synthesized, it is called as reverse transcription, and that is the modification of central dogma of protein synthesis. I think you have understood whatever I have explained you today. It is a very basic concept of molecular biology, and it is unique that how proteins are synthesized, because proteins play many functions in the biological processes. We know that all enzymes are protein, all hormones are protein, every metabolic reaction is possible in a body. In the absence of protein, the reactions are not possible. In the absence of enzyme, these reactions are not possible. Okay, so I think I will stop here today. Thank you.